First of all, thank you, the organizers, for the invitation. My name is E Eguiarte, and I work in the National Autonomous University of Mexico in Mexico City, around here. And here is a view of my institute. And I'm going to talk about the biodiversity in Mexico, how we are studying it, and two examples, one on the using the Cucurbita genus and another with the agaves. And Mexico is a country that has a very large diversity. About 10% of the biodiversity of the planet is found in Mexico, and plants are not an exception. In Mexico, we have about 30,000 plant species, and we have less than 10% of the biodiversity of the planet of plants. But the important thing is that a lot of plants were domesticated in Mexico, and good examples are maize, for instance, pumpkins, beans, and several other species. But not only this, about 100 domestication, but we also have a lot of plants that are very useful, and people still collect them from the wild. Here I have a table about medicinal and edible plants that are, most of them not domesticated, but are still used in Mexico. And I'm going to talk a little bit more about the plants of the traditional management of land in Mexico that we call the milpa. So in the milpa, we have the corn, the maize, and growing around the corn, we have the different species of beans, and in the soil, covering the soil, grow the different pumpkins. And here we have the archaeological data. So the domestication, we have good archaeological data for more than 8,000 years ago. And not only the beans, but also the pumpkins, the chiles we have in this figure, but also the agaves. These are the data from a cave near Tehuacan that indicates that agaves were also used by the old Mexicans from a long time ago. And the other thing that I want to remark is that in Mexico, not only we have the domesticated species, but also we have in many cases still the wild relative of the domesticated species. So we have here the teocintle, the wild relative of the corn, the chilpetin, the wild relative of capsicum, in this case capsicum annum. There are different beans, wild beans, in this case Phaseolus vulgaris. Here we have also the cotton, but I also put here the pumpkins, the wild pumpkins and the domesticated pumpkins. And not only that, we have a lot of land races still growing in different parts of Mexico. And this is the map of central Mexico. Here is Mexico City. And this is what we consider the southern part of Mexico, in particular the coast down to Central America, Mesoamerica. And in Mesoamerica, here still we have the populations of the wild relatives of the pumpkins and the beans and maize. So first I'm going to talk about agave. Agave, sometimes called in English century plants, are very important for ecological reasons in Mexico, but also for economic and traditional uses also in Mexico. And here we have the pollinators, the bats, mostly a genus of bats called Leptonicteris, and the process of cooking the central parts of the agaves. So here is the agaves, a little bit on the diversity of the agaves and the important things about the agaves, because they are very common. We have more than 200 species. Most of them are found in Mexico, and they thrive in very dry conditions, so they have interesting adaptations, and they have the largest inflorescence in the plant kingdom, perhaps up to 16 meters, are pollinated by bats. But for this talk, the most important thing is that they are used by people for drinks, that I'm going to talk a little bit about pulque, mezcal, bacanora, tequilas. It's still used for food, and it's important for fibers, and now there is a lot of interest in biofuels. So pulques, here we have some pulques agaves, and the whole goodness of the pulque. And pulque is just the sugars that accumulate in the central part of the rosette that are going to feed in a natural conditions to produce the inflorescence and the nectar, and the people extract and ferment it and is the traditional alcoholic drink of central Mexico. But now there is a lot of interest in mezcales and tequilas. Basically, a mezcal is any distilled beverage derived from the agaves, and tequila is a specific kind made with only one plant, Agave tequilana. And we have a huge diversity of mezcales. And also it's interesting, the name mezcal is how the people in the countryside in Mexico talk about the agave plants. They don't say usually agaves, they call them magueyes or mezcales. And here we have the central mature part of the agaves, that all this is cooked to produce the tequilas, the mezcales, but also they have a high content of sugars, and this head can weigh almost half a ton, 471 kilos in this case. And here we have some of the diversity of the agaves used to produce mezcal, at least 21 species, but I think up to 100. And I'm going to talk a little bit more about Agave angustifolia, that is here, that has a very wide distribution, not only in Mesoamerica, but also in the arid parts of Mexico, and Agave tequilana that grows mostly around the town of Tequila. And well, agave is a huge genus. We consider that it has perhaps almost 250 species if we include the herbaceous lineages. And here is the distribution of the genus in the Americas. Most of the species are found in Mexico, but some species are found in southern United States and in the Caribbean islands and in Central America. And here are the different states in Mexico and the species that we know that grow as native species of agave. And the states with highest diversity are Oaxaca, that later I'm going to return, and Jalisco. And yes, the agave genomes are very large. And for a while it was difficult to genome them, but recently two different groups, Alfredo Herrera Estrella in Langeb in Irapuato, and Xiaohan Yang in the Oak Ridge National Laboratory, were able to sequence, and they are working on the papers. And for agaves, the important thing I wanted to say is that in a report in 2001, they found no genetic variation. So that would be very dangerous for these cultivated plants. And also, in general, in the agaves, there is little genetic variation in the cultivated plants. So we analyze, again, the populations of tequila plants, but using next generation marker, GBS, and we found that the levels of genetic variation are, compared to other agaves, low, but nevertheless, we detected some genetic variation, very little genetic differentiation, and an excess of heterozygotes individuals. And we work with similar methods in some other populations in the north of Mexico with Agave pacifica that is used to make another drink, bacanora, and we found, yeah, about the double of genetic variation, very little genetic differentiation that I show here. Basically, the wild and cultivated plants are the same, and very little genetic differentiation. Here is the coast. The coastal populations are different from the highlands populations, and very low clonality. So yes, we found a reduction of genetic variation, and we think that that can be very important for the future of the populations. This happens because the agave can be propagated clonally, and we will need to pay a lot of attention to preserve the genetic variation of this genus. We have been also working with pumpkins, and this is the group of people that has been collaborating with me. Pumpkins is a small genus, only 20 species, but this genus was domesticated at least 6 times, most of the times in Mexico. And here is just to view a little bit of the morphological diversity in the genus. And again, some of the morphological diversity and the giant pumpkins that can be selected. And here is the phylogeny. And I just wanted to show where are the different domestications, the differences in the size of seeds. And yeah, the genus has evolved from the northern Mexico to the more tropical regions. And I'm going to very briefly concentrate in this clade. Cucurbita argyrosperma. We have been doing analysis both of the genomes and of the population genetics of this group of species. And basically what we found is that if the cultivated plants still have a lot of genetic variation, we assembled the genomes at the chromosome level, and we discovered that it was apparently domesticated in the lowlands, again, of Jalisco. And using coalescent models, we discovered that it was domesticated perhaps down to 14,000 years. And we have been using these data and genomes to reconstruct the history of the species and to find the different genetic markers that are associated with domestication. So we think this advance is very important, and we are going to use similar methods in agaves. And this way we can use molecular markers to improve the populations, in particular in face of the global change. And with that, I just wanted to thank all the funding agencies, especially the CONABIO that you saw the logo several times. It has funded a lot of studies on domestication of plants, in particular our Cucurbita studies, and the National Science Foundation of Mexico, CONACYT, and a lot of people that has helped us in the field, especially the people that domesticated and still have these genetic resources. And thank you, everybody, for your attention.